Good morning. This work was done completely during the lockdown. We were at LMU in Munich, Germany, in sabbatical, and at the beginning of March, we found ourselves completely disconnected by our country, families, and friends. And we started to think how we could help our country. We are scientists. My name is Caterina La Porta. I am pathologist and I work on quantitative biology, system biology, and network medicine. And I'm teaching immunology for at least 20 years. And with me was Stefano Zapperi. He's a theoretical physicist working on complex systems and a statistical physicist who just received an Humboldt Research Award. And we are also a family. So what was our question? Our question was, why SARS-CoV-2 infection leads to severe symptoms only in a fraction of patients? To answer this question, we considered HLA-1. HLA-1 is a molecule that is expressed by all nucleated cells, and his job is to present intracellular peptides that are produced by proteasome activity to the T cells in order to induce an immune response. An important point to keep in mind is that HLA-1 has a small pocket that allows to keep inside peptides that are between 8 and 11 amino acids. And in general, another important point is that HLA is a polymorphic molecule, and we considered in our paper a subset of HLA-1 that represents 90% of the entire human population. So what was our idea? To reproduce in silico the machinery of a cell infected by SARS-CoV-2 expressing HL1 SARS-CoV-2 peptides. And here you can see a small summary of what we got. First, we computed the binding of SARS-CoV-2 peptides to HLA molecules. We started to investigate the individual immune response to SARS-CoV-2, and we identified weakly or strongly binding haplotypes in human populations. So, starting from the first point, we considered, in order to get these results, we considered the more expressed proteins, viral proteins in infected cells, according to the paper published in 2020 on eLife. And what we did is to reproduce, to simulate in silico the proteasome activity. Proteasome activity works degrading on a specific point of the proteins. The proteins that arrives to proteasome should be targeted by ubiquitin, and as a result, you have many different peptides. So we reproduce in silico exactly all these activity that you see in this picture, and as a result, we got peptides of the virus. We selected the peptides for the sites, like the cells do, exactly the same as they used to do. So we selected peptides between 8 and 11 amino acids. And then we used a neural network to estimate the binding affinity of 130,000 HLA-1 peptides. And here you can see the results. In this picture, you see a ranking of the different alleles of A, B, and C of LH1 and the number of peptides. So on the top, you see alleles that are weakly binding. They show very weakly binding with peptides, and on the bottom, strong binding. And you see that we compare also SARS-CoV-2 with SARS-CoV and with the coronavirus that is involved in normal cold. And you can see that they show closer similarity. Second point that we explore is try to explain the variation in immune response in the study when an HL1 binds with strong or weak capability a specific peptide, if this is important for the response, the capability to induce a response in T cells. So what we did is shown in this picture. So panel A and B shows a ranking of HLA alleles, different HLA alleles, with respect to peptide stability and T cell propensity. We put together these two algorithms in the combined score, and you can see the results in the C panel. And you can see that when specific HLA binds a specific peptide with strong capability, with a strong binding, this means also a higher T cell propensity, so a higher capability to induce a T cell response. The third point was to identify, to study better what happens across a human population, because it's known that HLA haplotypes are distributed differently across the population. And so we checked the interaction of each HL1 peptide with strong or weak affinity in different subpopulations. And you can see in this picture that across a human population, individuals that have weak or strong haplotypes are different. So our conclusion is that our strategy can help to identify high-risk subjects. You can read, of course, more details and data on our paper appeared on Cell Systems in 2020. Thank you so much for your attention.